So for the health science course, evidence-based research methods, we have to write a systematic review, and I chose to write mine on the efficacy of favipiravir as a form of treatment for two different filoviruses, and those were specifically Ebola and Marburg virus. And I kind of started this by researching different science databases such as like PubMed, JSTOR, PLOS journals, ProQuest Central, and Science Direct. And I searched for randomized controlled experiments of favipiravir's effect on Ebola or Marburg virus. I was able to limit the scope of my topic in between September and October of 2019. And I was writing my paper in October, so I kind of needed like a clear cutoff date where I didn't have to include any research published while I was writing my paper. But also, the majority of these studies were published after September just because it was shown that favipiravir was reducing the viral load of influenza in a bunch of different test subjects, and they were kind of trying to see if this was a universal effect and if we could use favipiravir as a form of treatment for these more high mortality rate viruses, such as different filoviruses. So all the papers I included were peer-reviewed primary studies that focused on favipiravir as the sole form of treatment for either Ebola or Marburg virus. So if these studies had favipiravir as a form of treatment in conjuncture with another treatment method, I excluded the paper just to kind of keep my scope limited. Additionally, if they weren't a small animal model or a non-human primate model, I excluded the study. There was a single human trial done during the Ebola epidemic in Africa, but there was just a lot of confounding variables to this trial, and the results were really inconclusive, so I just kind of exclude that. I do talk about it a little bit at the end, but for the most part, I just focus on those small animal models and the non-human primate models. So favipiravir works to inhibit viral RNA polymerase during the transcription process, and it does this by acting as a synthetic guanidine nucleoside analog. So if you look at figure 1, you can see the chemical structure of favipiravir compared to the chemical structure of guanine, and you can see how similar those are. So basically, as RNA polymerase is transcribing DNA to mRNA for the virus, it will pick up favipiravir and insert it in the place of guanine, and this basically acts as an early stop codon and cuts short these mRNA strands. And this really helps reduce the total amplification of the virus within these subjects, and what you saw was a reduced overall symptoms that the virus actually would exhibit, and this was really helpful in reducing the overall mortality rate, especially in filoviruses, which basically kill their host by inducing hemorrhagic fevers. So the majority of the models I looked at were small animal studies, and they reported treatments around 300 milligrams a kilogram a day, including groups that were dosed twice daily, had about 100% survival rate as long as they were injected with favipiravir in a certain amount of time post-infection. Within all these studies, if those parameters were met, they only had one mouse die from this. So the common symptoms seen in these treated mice were weight loss and slightly elevated body temperatures on day 8, but they never reached the hemorrhagic fever stage, which was what ultimately kills most of these infected individuals. So the mortality rate plummeted. Additionally, favipiravir worked to repress the replication and reduced the viral titer by about 50% in these small animal models. Figure 2 is going to show a bunch of the different results seen from Madeline et al.'s research. So in that first column, you see the no treatment graphs. In the second column, they have a graph from a group that was treated with 150 milligrams a kilogram a day of favipiravir starting at day 6 post-infection. And the third column is going to show the exact same dosage, but this started at day 9 post-infection. So kind of looking at the rows, the first row is going to show the survival rate of the different treatment groups. And you can see that there was a 100% mortality rate at day 10 with no treatment intervention. And in the third column, you're going to see that while these mice did live to day 15, they were all dead at that point as well. What's really interesting is if you look at the second column, you can see that if the drug was administered within 6 days post-infection, they had a 100% survival rate, which was a massive finding. Additionally, the next row down shows the total weight loss, and while column 1 and 3 show a loss of weight with no recovery, column 2 shows that while there is an initial dip, as soon as they're treated with favipiravir, you start to see that weight climb, and they actually ended the study with a higher weight than when they started the study. And row 3 is just going to show the total viral load, and you can see that when favipiravir is administered 6 days post-infection, it never reaches that maximum load that causes the hemorrhagic fever, as you see in columns 1 and 3. So they're really thinking that's why these mice were able to develop the antibodies necessary to fight back these diseases. So while most of the small animal models had similar findings to Bladholm et al., the non-human primate models were a little less conclusive in their results. They did show that there was an increase in survival rate to about 40 and 60% if they were dosed properly within a certain amount of time. But one of the other studies showed less positive results, and they're not sure why that is, if it was because of the strand of Ebola they used, or if it was a confounding variable that they're unsure about, or just kind of that kind of stuff. So they need to do additional research into this. Additionally, they found that favipiravir needs to have an active kinase present to phosphorylate into its active form. Several of the studies noted that the amount of the active form differed from tissue to tissue. However, this didn't seem to be a huge issue in how favipiravir worked to reduce the overall viral load, just because in the states or the tissues of the body that had a higher amount of the virus also had a high amount of that kinase, and so they did see a high concentration of the active form of favipiravir. And that whole mechanism is kind of shown in figure 3, along with several other different treatment options that they found that work in a similar manner as favipiravir. So just kind of as like a brief summary, while Ebola viremia did see about 50% reduction in the small animal models, the non-human primate models were a little less conclusive. Marburg virus also saw a pretty similar reduction in the small animal models. However, one of the non-human primate models showed a 40% survival rate, which is significantly higher than the 8% survival rate reported in the other non-human primate study. So they're not quite sure what caused this difference. One of them was an oral and the other was an intravenous, but that didn't seem to make a difference in the small animal models. So they just need to do some more research into that. Additionally, the Jiki trial showed lower viral titer, as well as a significantly lower mortality rate, but like I said, those confounding factors really made the results of this test inconclusive, so they just need to do a lot more testing, which is really hard with this kind of topic. But at the end of all this, favipiravir could be a really good method of treatment for filoviruses, but there might be better methods out there that we need to research. Thank you for listening. I hope I wasn't too boring, and I hope you guys have a good surf day.